In this video, we will discuss section 10.2 on the anatomy of skeletal muscle. Each skeletal muscle is an organ in itself. We have multiple types of tissues going on here, and they're all going to work together. We've got our skeletal muscle fibers, we've got connective tissue in between here, we've got our blood vessels and nerves coursing through as well. When we have our muscle fibers bundled, we call that a fascicle. So we have many fascicles in here. This is representing a fascicle. Here we have another fascicle, and in here we have the muscle fibers. And our whole muscle is going to contain many of these fascicles. When we look at those fascicles, like I said, we can see they're muscle fibers. So here are bundles of muscle fibers, and here's one pulled out, and each of these muscle fibers serves as a muscle cell. So we'll be using those terms interchangeably here. And we need to talk about some connective tissue compartments that our muscle has. We have 3 concentric layers of wrapping. We're going to start more superficial. So around the entire muscle itself, we have something called an epimysium. Epi meaning upon, myo meaning muscle, and eum meaning pertaining to. So this connective tissue wrapping is made up of dense, irregular connective tissue. Next, moving in deeper, we have a perimysium. And this perimysium is also made up of dense, irregular connective tissue and will serve to wrap around each of the muscle fascicles. We'll also see within this perimysium, we have lots of these blood vessels and nerves coursing through to serve the muscle. And lastly, the innermost connective tissue compartment is going to be an endomysium. Now here we have our connective tissue changing. It is made up of loose areolar connective tissue, and we'll see that it is wrapping each of these individual muscle fibers or muscle cells in here. This is just going to be a delicate layer, and its main function is for electrical insulation, capillary support, and binding to neighboring cells. Now let's talk about some attachment points of muscle. When we have a muscle that is attaching to a bone structure, this is called a tendon. This is a cord-like structure, and it's made up of dense, regular connective tissue. Notice how with the epimysium and perimysium, this was made up of dense, irregular connective tissue. Now, we've talked about aponeuroses in class. This is when we have thin, flattened sheets of dense, irregular tissue. Really, it's a flattened tendon, and we could see this best with our external abdominal oblique muscle or internal abdominal oblique muscle, and even our transversus abdominis muscle that lies below this internal oblique. All of their tendons are thin and flat, and they come together and form this aponeurosis, which we call abdominal aponeurosis. Next, let's talk about fascia. So we have 2 layers of fascia. If we look at the top or superficial portion of our body, we're going to see, of course, our skin, and then we have a superficial or adipose tissue layer. And this is going to be right above where we find our superficial fascia. Now, if you look at this view down here, this is a cross-section through the arm. And we could see the skin out here, and then where we see adipose tissue here, that's serving as the second layer. And then where we see this thin bluish purple line, that is our superficial fascia. This is made up of areolar or adipose connective tissue. And of course, we'll find this superficial to the deep fascia that we'll point out in just a moment. And it really helps to separate out our muscle from the skin itself. Now, we can see the muscle underneath here, and our muscle is going to be separated from one another within the arm. And so the connective tissue layer that separates out those muscles are called the deep fascia. And this is made up of dense irregular connective tissue, and we find it just superficial to that epimysium. So let's say here is the, I don't know, let's say the lateral head of the triceps brachii. Then we would have the epimysium that surrounded that muscle first, and then more superficial to that, we would have this deep fascia. We'll also see that the deep fascia is going to help bind the muscles with similar functions. And so if we go back to this layer that we see here, that deep fascia can be found just above the muscle here. Now, how is our muscle tissue vascularized and innervated? Our blood vessels are going to highly vascularize our muscle tissue, meaning we've got lots of blood vessels around them and lots of capillaries, because our muscle cells are going to need oxygen and nutrients so that they can function properly, and we need the blood vessels, mainly veins, to be able to carry away the waste products. Then our innervation is going to be taken care of through our somatic motor neurons, meaning we are going to have nerves that start off here in the brain. These are known as upper motor neurons that will travel through and then synapse somewhere within the brainstem or within the spinal cord with a lower motor neuron that will move on out to go and supply the actual skeletal muscle. Now here's a little bit closer view of what that looks like here at the spinal cord level. So we'll see the cell body of our neuron that we're going to learn about soon exiting. There's my axon of my nerve traveling out the spinal cord and then moving through the body. And here we see the terminal of that nerve. And this area here is called the neuromuscular junction, because I've got my nerve, I've got my muscle, and it's at a junction, right? And this nerve allows for voluntary muscle contraction. Really, our central nervous system is initiating this within the motor cortex or cortices, and then allowing us to say, hey, I want to pick up that pencil, so that we're going to move down to the spinal cord level that's appropriate and go over to our arm or forearm muscles to allow us to do that. Now let's talk about the parts of a muscle cell or muscle fiber. So we have renamed parts of the cell, of the muscle cell, I should say. Instead of calling the cell's fluid portion that contains all the organelles a cytoplasm, we are now going to call it a sarcoplasm. Sarco means fleshy, and plasm is remnant of cytoplasm. So this is just our fleshy cytoplasm really here. And this is the area where we're going to find the organelles of our muscle cell and our contractile proteins that we're going to talk about in just a little bit. And they also have some other specializations that go along with them. Another thing that's really special about our skeletal muscle fibers is that they have multiple nuclei. And that's what we mean when we say multinucleated. So if you look at these full skeletal muscles here, skeletal muscle cells, we see that these purple dots here are representing the multiple nuclei. And the way that we are forming this skeletal muscle fiber is when we are in embryo, we will have multiple myoblast cells fused together. And some of these nearby myoblasts are going to become undifferentiated and become satellite cells. So we really don't have satellite cells shown in this photo, but they're literally just cells kind of hanging out here like a satellite. And they help to support the muscle and will be there in case any of our muscle fibers need any repairing. Now for another special term, our sarcolemma is the term that we are going to use for our muscle cell's plasma membrane. So this in like the translucent color here is serving as our sarcolemma. Notice how it's forming a boundary for our muscle cell. And this is really going to be important because our sarcolemma has a voltage-gated ion channel that's allowing for conduction of electrical signals here. So we're going to see that the signal will travel through and then send it through other specialized structures, like the T-tubule here, for example. And that's what we're going to talk about next, are these green lines that are representing our T-tubules. And I'll show you in a different photo as to why these are called T-tubules or transverse tubules. We'll see that a portion of them will kind of go across to make contact with the sarcolemma, and then it moves down, being like this bottom portion of the T. So that's why they were called that, and they go deep into the muscle cell. And we'll see that they also are going to contain important voltage-sensitive calcium channels. And here's that photo I was talking about. So over here we have the sarcolemma, and notice how we're having changes in ions here. That's what all this positive and negative means. Here we see we have a voltage-gated sodium channel that's bringing sodium in. Here's a voltage-gated potassium channel allowing potassium to move out. And it's moving down this green portion, which is our T-tubule. So in that manner, we're allowing electrical conduction to move down the T-tubule. So almost imagine this is the top of my T, this is the bottom portion of my T. So transverse tubule. And as we move down, we can see that calcium channel I talked about in the previous slide. That is going to link over to what we will learn about in just a second, this sarcoplasmic reticulum that contains all this calcium in here. First, we need to define myofibrils. Our myofibrils, I know this is a little tiny in this photo, but here is our muscle fiber. I don't want you to get confused on terms here. And inside that muscle fiber, we have these myofibrils. And again, another confusing term here. Inside this myofibril, we have many myofilaments. So here you can see one of these myofibrils pulled out, and you could see these tiny myofilaments inside of them. Now, let's go back to that sarcoplasmic reticulum term. So the green we said were our T-tubules, in which we are going to send that electrical impulse through. In that T-tubule, we had a calcium channel, and that was going to allow that electrical signal to tap into the sarcoplasmic reticulum, which, by the way, if you didn't gather, this sarcoplasmic reticulum is a new name for a cell's smooth ER. And we have two different ends of it. These portions in here are known as the terminal cisternae. So I'm going to go back one slide to show you this. So here's the sarcoplasmic reticulum over here, and the portion that is closest to this T-tubule, this is called the terminal cisterna, or if we're talking about plural, terminal cisternae. And so on both sides, the terminal cisternae and the T-tubule in the middle is all known as a triad. Okay, so one terminal cisterna plus T-tubule plus another terminal cisterna of the sarcoplasmic reticulum is known as a triad. Now these cisternae serve as reservoirs for calcium. So if I need to find calcium, I'm going to look inside the sarcoplasmic reticulum, especially here at the terminal cisternae. This is where the calcium is going to be released. Once the T-tubule senses that impulse moving down, this is voltage sensitive channel, right? So that voltage moving down is going to say, hey, calcium channels, you need to release this calcium over here into the sarcoplasm. And we'll talk about why in just a minute. Also, those calcium pumps that are going to import the calcium is going to bind to something called called calmodulin and calsequestrin. Now let's talk about the myofilaments. These are going to be those contractile proteins that we saw in the previous slide, right? So here we have our muscle fiber, our muscle cell. Inside there we had our myofibrils, and then within the myofibril we have our myofilaments. And we have two different types. We have these thick filaments, which are also known as myosin filaments. You could see that we have bundles of the myosin protein molecules. So this is just one myosin protein molecule here. We have the tail end, and this would be the head. It almost looks like a boxing glove over here. And we have these special actin binding sites and ATP binding sites. And we'll discuss that a little bit more later on. But it just almost looks like you piled a bunch of golf clubs together in order to make this thick filament here. And then when we look at our thin filament, our thin myofilament, also known as our actin filament, you see that we have twisted strands of actin. So we have really two terms for actin. We have F-actin, in which we're talking about this longer strand portion, and our F-actin is composed of these little ball-like structures which are known as G-actin. So G-actin, you can kind of think of more single units, whereas the F-actin, think of a bundle of these G-actin proteins. So our G-actins, I always think of them as little blueberries because if you look at this picture carefully, you have that little opening, and these serve as myosin binding sites where we're going to see these myosin heads will attach, specifically at the actin binding site here. We also have some special proteins associated with our thin filament here. We have tropomyosin, which is in the green, and I like to think of that as kind of like the jump rope of this myofilament. It's a long word, and I just think of a long jump rope. And then the other protein we have is troponin. So troponin kind of reminds me of the word triple, and we've got 3 subunits here that make up troponin. And notice how it has a special binding site on here for calcium. So before we go into organization of a sarcomere, I want to play a little video that summarizes what we just talked about.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open.
So remember that our sarcoplasmic reticulum contains the calcium, so that's what we want to come out into the cytoplasm to move down to our thick myofilament, which is the myosin. Well, not really the calcium to that, but we want the calcium to go to our thin myofilaments here, or the actin filament,
resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on 2 sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament.
So this information is going to be new, but I think it'll be a nice little preview into the coming slides.
Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
So now let's jump back into our slides here where we're going to talk about a sarcomere. So remember, here is our myofibril that contained the myofilaments, and notice that each of these or not each of these, but within our myofibril, we have many sarcomeres. And they are going from this zigzag line to another zigzag line. And these zigzag lines are known as Z-discs. So like I mentioned in the beginning, this sarcomere is our functional and structural unit of our muscle, our skeletal muscle. So we will have repeating units of these sarcomeres that make up the myofibril. And in here we can also see our myosin filaments and also those actin filaments. A little bit harder to differentiate them in here, but we will look at a magnified view of this. These Z-discs, let's come back to those. And I'm actually going to start referencing this area in here because it is going to label all the different parts of a sarcomere for us. These Z-discs are specialized proteins, and we're going to find that it is perpendicular to those myofilaments that are parallel to one another. It also is going to help to anchor our thin filaments. And the positions of these thick and thin filaments are going to give rise to alternating I bands and A bands, which we'll talk about on the next slide. But notice how the I band portion here is lightly colored, whereas the A band portion is dark. So here we are. Those I bands, like I mentioned, are the light portions, and that's because they are only containing our thin filaments, those actin filaments, which makes sense because there's our Z-disc holding those thin filaments in place. And we'll see that this area will become smaller as the muscle contracts and can disappear if we have maximal contraction. So I'll show you that in another slide when we look at a sarcomere contracted. Now for this A band, we said this was our dark appearing region. And in this area, we're going to see thick filaments overlapping with the thin filaments. And that's what gives it this dark pigmentation. You could see that in the center here, we have a lighter area, right? And this is the H zone. Within the H zone, and I'm just going to fast forward here so you could take another look at this really colorful picture. But here, let's point out the A band again. This was our dark portion. And here we can see the overlapping thick filament and thin filament portion. But this area that was the H zone, that's light in color. And that's because we don't have the thin filaments overlapping with the thick filaments here. So that's why this center portion seems a little bit lighter. And then in the center, we have what's called the M line. And this is another thing that is anchoring for our filaments, specifically the thick myofilament here. As this sarcomere is going to contract, we'll start to see these thin actin filaments move toward the M line. So what's going to happen is this H zone is going to become smaller or possibly disappear as we reach maximal contraction. All right, now let's move into that H zone that I discussed. I think I mentioned everything here. And I also mentioned our M line. It is made up of proteins. And like I said, those thick filaments will attach there. I just get ahead of myself a little bit. And then I just wanted to show you in this view the I band too. Notice how we have our Z-disc in the center here and only our thin filaments, which is why this is going to be lighter in color. And here is a little bit different view that shows you your sarcomere in a cross section. So here is Z-disc to Z-disc. That's our sarcomere. We always measure our sarcomeres from Z-disc to Z-disc. And here is an M line. And so we're just a little lateral to that M line, and we have sliced this cross section there, and you could see that we only have thick filaments in that H zone area. And then if we move a little further out, this is where we have our A band because our myosin filaments and our actin filaments are overlapping one another. So that's what that looks like. And lastly, as we get closer to that Z-disc, we can see our I band. And when we look at this in cross section, we could see only our thin filaments And we are going to talk about connectin. That's what's pictured here in the green along with those thin filaments. And here it is, connectin. This is going to extend from the Z discs to the M line. So you kind of can't see the complete attachment here to the M line, but what this is in place for is to stabilize the thick filaments, and it is coiled up because it has a spring-like property, and this will assist us in passive tension. Another really important component of muscle is dystrophin. And you can see our dystrophin over here in this photo. Here is our sarcolemma, and we have some other components here that we aren't going into detail with, but notice how it is attaching to some of our myofilaments. So this dystrophin is really important to anchor some of these myofibrils to our sarcolemma. And then sometimes there are abnormalities within this dystrophin protein, and that can cause muscular dystrophy. And let's talk about muscular dystrophy. This is a collective term that we use for hereditary diseases where we see skeletal muscle degeneration. And so we have to mention Duchenne muscular dystrophy, or DMD, because it is the most common type. We see that within this disorder, there's defective or insufficient dystrophin, that protein that we just talked about. We'll also see sarcolemma damage during muscle contraction, and the calcium enters the cell and damages the protein. The problem starts in early childhood. We start to see that there are walking difficulties. The knees may bend back to take weight. We have thin, weak thighs and poor balance, or they may be very clumsy or just walk awkwardly. And we'll see that we have thicker lower leg muscles and a tight heel. So you might see that they are walking on their toes, kind of like that ballerina drop. And also in certain areas, we will have weak muscles, such as the anterior leg muscles. So we will have something called foot drop that can take place. We'll see in other areas, there will be muscle atrophy, where the muscle is shrinking. And of course, posture issues. They will have a noticeable increase in their curvature within their lumbar vertebrae, and that'll cause something called sway back. And unfortunately, this is incurable. Patients rarely live beyond age 30, but there is a lot of research being done within this field in order to slow muscle atrophy and assist these patients as best as possible. Now let's talk about some of the other structures we find within our muscle cell. We are going to have lots of mitochondria in here in most of our skeletal muscle, and that really is in place for aerobic ATP production, which is going to call upon oxygen. So we'll get into that a little bit more later. But another thing we need to mention are these myoglobin molecules. These are within our cells and will allow storage of oxygen so that we can use it during aerobic ATP production. So we'll see this in more of our slow twitch muscle fibers, which we'll get into later. Another thing that's important within our muscle cell is that we are able to store glycogen. So we call that muscle glycogen, and we are going to be able to take glycogen and break apart some of the bonds in order to get glucose so we can use it for glycolysis. Another item that we need is creatine phosphate so that we can quickly give up the phosphate group in here and use it to create ATP. Let's move into innervation of our skeletal muscle fiber. Our muscles are given innervation through motor units. This means that from our spinal cord, we are going to have a motor neuron, or sometimes we can call this an efferent neuron, that will travel to these muscle cells or muscle fibers and help to control it. These axons are then going to work to innervate a lot of these muscle fibers. You could see them going to different muscle fibers, and the number of fibers of a neuron will innervate a different number of these muscle cells. So you can see that this red axon has moved on and branched to supply this muscle fiber, this one here, and here. So we've got 4 of these muscle fibers supplied by, I'm not sure what that line is pointing to, maybe axon motor neuron 2 in the red. And the blue, the axon of motor neuron number 1, is just going to these 2 muscle fibers. So we'll see that our smaller motor units will have less than 5 muscle fibers, and that allows us to have more precise control for force output. So this could be for, let's say, our eye muscles or maybe our smaller muscles of the hand, whereas our larger motor units are going to go to thousands of muscle fibers, and that allows for production of a large amount of force but not precise control. So this could be like a motor unit that is going to erector spinae or quadriceps femoris. And our fibers of our motor unit are dispersed throughout the muscle, so they're not just clustered within one compartment. Now let's talk about that point where this neuron is close to the actual muscle cell. Remember, we said this was called your neuromuscular junction. This is where the motor neuron is innervating the muscle itself. And this is usually going to be about the mid region of a muscle fiber. And we've got a couple different parts to this. We've got our synaptic knob, which is this end portion or like this swollen area of the end of the nerve. Then we have a synaptic cleft, which is the space between the synaptic knob and the muscle. And then just to make this a little clear, I don't know if it's going to let me do this. Hopefully it does. But I want to make it clear what the motor end plate is. It's this portion over here. Notice how it's still my sarcolemma of my muscle cell, but it has a lot of these invaginations, and that helps to increase surface area. And then we see the sarcolemma kind of clears on out here. So if we take an even closer look at that neuromuscular junction, we see that there is a lot going on. Where we have this synaptic knob, again, it's this expanded tip of the motor neuron axon. We're going to see in here these spherical containers, and these are called synaptic vesicles. And these synaptic vesicles are filled with a neurotransmitter called acetylcholine, or ACH. In order to get this neurotransmitter out, we need an action potential to move down this to the end of this nerve or this axon, and that will trigger this voltage-gated calcium channel to open up, and that is going to have calcium move in. Now, when calcium moves in toward the synaptic, inside the synaptic knob, we're going to see it bind to the synaptic vesicle, and that causes this vesicle to move up toward the end of this synaptic knob and exit through exocytosis. So notice how we now have released acetylcholine. And what we want acetylcholine to do, if you follow these red dots, is bind to an acetylcholine receptor. So that's what this gray colored structure is here. It's an acetylcholine receptor. Once that acetylcholine receptor, I might be getting ahead of myself here, yes. Once this acetylcholine receptor has its acetylcholine attached to it, it's going to allow for sodium to move into the cell and potassium to move out of the cell. So we've got lots of ion exchanges taking place here. If we look at our synaptic cleft here, this space, we see it is fluid filled and it's separating this motor end plate from the synaptic knob. Now in this area, we are also going to have something called an acetylcholine esterase. and that is going to break down acetylcholine when we're done using it. Now let's take a look at an animation of all of this taking place.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So in that video, you saw the term action potential mentioned, and that's what we're going to talk about next. Our muscle fibers, when at rest, are going to exhibit a resting membrane potential. We've got fluid inside the cell, and this is going to be negative when compared to the fluid outside of the cell. So inside our muscle cell, we have a voltage of about negative 90 millivolts. And our resting membrane potential is going to be established by some leakage channels, which are not pictured in this photo, but just imagine they kind of look like this, except they don't have this little closed part to them. And these leakage channels are going to be for sodium and potassium, so they'll just flow across their natural gradient. And then we also have something called a sodium-potassium pump. Now let's review what a sodium-potassium pump is.
The sodium-potassium exchange pump is an example of an active transport process. Active transport processes can move substances against a concentration gradient, and they require energy expenditure in the form of the breakdown of ATP. The sodium-potassium exchange pump moves sodium ions out of the cell and potassium ions into the cell. The carrier protein for sodium-potassium exchange is embedded in the cell membrane. 3 sodium ions and an ATP molecule can bind to the carrier protein on the inside of the cell membrane. The ATP is broken down to ADP and phosphate. Simultaneously, the carrier protein changes shape and the sodium ions are transported across the membrane and released on the outside. In its new conformation, the carrier protein can now bind potassium ions on the outside of the cell. 2 potassium ions move into the carrier protein from the outside of the cell membrane, and the phosphate attached to the carrier protein is released. The carrier protein changes shape again, reverting to its original conformation, and delivering the potassium ions across the membrane to the inside of the cell. It is now ready to repeat the process of sodium-potassium exchange.
So by use of that sodium-potassium pump, we are going to be pumping potassium into our cell and sodium out. So majority on the extracellular side will be sodium, and the majority on the inside of the cell will be potassium. And at this point, at rest, we'll see that these voltage-gated sodium channels and the voltage-gated potassium channels are closed. So let's take a look at what is happening here. We have our neuromuscular junction that we talked about. Acetylcholine was released to these receptors that allowed sodium to move in and potassium to move out. So we just talked about how at rest, the inside of our cell is negative 90 millivolts. Well, if we have all of this positive sodium moving into my cell, that -90 is going to start to what we call depolarize, meaning it's going to turn more positive. So we move from -90 millivolts to, let's say, -85 millivolts, -80 millivolts to -75 millivolts, and so on, until we reach a certain threshold, and then we get an action potential or depolarization to take place, meaning that when we're at rest, the membrane, let's say, is polarized. The inside is negative, the outside is positive. It's polar, right? So we're almost undoing that. We are depolarizing that as the inside becomes more positive and looks more like the positive outside. So that depolarization causes our action potential to propagate, meaning it's going to move down the sarcolemma, and then soon we're going to talk about how it moves down the T-tubule, and we'll see the voltage-gated calcium channels start opening up and allowing that calcium to leave the sarcoplasmic reticulum here into the cytoplasm and over to our actin filaments in order to cause a cross-bridge cycling. So that's a little summary before we move into section 10.3.